Good morning, good afternoon. We're back to you again with another broadcast from the Virginia State University's Cell Physiology Spring 2016 group. And today we'll be presenting you a paper on the cell cycle. The name of our paper is MIR 34A, and it's regulation on mesangial cells proliferation via the platelet-derived growth factor receptor beta and RAS-MAP kinase signaling pathway. I'm Anthony Fields, and I'm along here with my colleagues, and we'll be going through our presentation.
Hello, I'm Shantara Stewart, and I'll be going over the RAS-MAPK signaling pathway. So first, if you're looking at the picture to your right, it's showing the epidermal growth factor, which is the EGF, that binds to the epidermal growth factor receptor, which would be the EGFR, which is the RTK receptor, tyrosine kinase, which is activating the tyrosine kinase activity. The growth factor receptor phosphorylates on the tyrosine residues. Then the growth factor receptor bound protein 2, which is the GRB2, is an adapter protein that binds to the phosphorylated tyrosine residue of the activated receptor. The SOS, which is the Son of 7 list, it binds to the RAS, which is a membrane-bound protein causing the SOS to activate. The inactive RAS will bind to the GDP, which is a nucleotide, and that will promote the SOS to remove the GDP. The RAS will bind to the GTP, and that becomes active. So the RAS binding to the beta RAF, which is a effector protein, then will phosphorylate and activate the MEK1 and 2. And MEK is the nitrogen activated protein kinase. The MEK phosphorylates and then it will activate the ERK1 and the ERK2, which is the MAPK, which is the nitrogen activated protein kinase. That will then go on to activate the Jun and the Phos, which are transcription factors. And the Jun and Phos form a heterodimer and bind to the motif of the DNA. That will lead to expression of their genes and the growth factor, and cell proliferation is activated. So how this pathway relates to the paper we're doing is that MIR34A, which is the microRNA, which has 34 nucleotides. It's a growth factor, and that binds to the platelet-derived growth factor receptor beta. And the receptor protein will bind to the PDGF, which is the platelet-derived growth factor protein, which will then activate the RAS-MAPK signaling pathway. And downstream, it also activates the target genes and the cell cycle proteins like cyclin D1 and E, which will control the cell proliferation.
Hello, my name is Petrita Raditzis, and I will be going over the introduction of this paper. Mesangial proliferative glomerulonephritis is a disease of the kidneys that affects the glomeruli. It is divided into two forms: immunoglobulin nephropathy, which is the inflammatory form, and the non-immunoglobulin nephropathy, which is the non-inflammatory form. In each case, hyperplasia is exhibited in all or most of the globular mesangial cells, along with several levels of extracellular matrix accumulation. Reducing and inhibiting cell proliferation of mesangial cells is important in treating proliferating glomerular diseases. They use an experimental model for this study. The standard animal model for mesangial proliferative glomerulonephritis is the anti-THY1, which is a combination of THY1 antibody and THY1 antigen, which makes up THY11. It is a GPL-linked glycoprotein in rats. And it's also detected with the clone OX7. There were several mechanisms of the renal disease, mesangial proliferative glomerulonephritis. Glomerular damages occur when macromolecular antigen antibody complexes are formed in or on the site. They become deposited in the mesangial cell area. This leads to both mesangial cell proliferation and inflammation and stimulus of activation. Once this happens, shrinkage and mesangial cell proliferation occurs. Consequently, a variety of inflammatory mediators and ECM components are released. These events subsequently exacerbate inflammation. This results in further damage to the molecular barrier of the glomerular filtration membrane, along with protein in the urine or albuminuria. MicroRNAs are a single-strand RNA molecule. It's 21 to 25 nucleotides. In eukaryote, microRNA are post-transcriptional regulators. MicroRNAs do not code for proteins. MicroRNAs regulate proteins by inhibiting translation or degrading target mRNA. This is made possible by its ability to bind to specific target sites. MicroRNA is involved in many processes associated with diseases in humans, including renal diseases. For this study, they're going to use microRNA 34A because it's been found to be associated with tumor proliferation of many cancers through target gene regulation. The authors of this paper hypothesized that the purpose of the study was to clarify the role of microRNA 34A in renal mesangial cell proliferation.
So what I would like to first introduce to you is the initial table of the paper. The table shows the silent RNA sequence. Silent RNA sequences interfere with gene expression of certain genes. They function by breaking down mRNA after transcription, so there's no translation. And where they're identifying here is the target gene that they're trying to find, which is the platelet-derived growth factor receptor beta. The sense strand is the 5 to 3' end, and the antisense strand is the 3 to 5' end. And in this paper, they will be referring to it as the SI-con.
Okay, the next table for table 2 is the real-time quantitative PCR primers for these target genes. The purpose of this table was to look at the transcription levels for each of the genes. For each gene, you see that there's a sense and there's an antisense. Sense is the coding strand. Like I said previously, it runs from 5' to 3', and antisense is the complementary strand that runs from 3' to 5'. And you're seeing the base pairs for each fragment. Okay, the next table, table 3, is the PCR primers for the 3' UTR fragments. And these are the targeted genes of the platelets, it's the platelet-derived growth factor receptor B, well as beta, and that's the CDKS would be the cyclin-dependent kinases 2, and then cyclin E. And the purpose of these target genes were to be amplified from the rat mesangial cells by PCR. And it's targeting the actual cDNA, which is the complementary strand, so this is looking at the antisense. And each fragment is containing the sequence for the MIR34A, which is the growth factor.
In this figure, figure 1A, they found that Ki67, a protein that is exclusively associated with cell proliferation, they found it during the study. On day 1, they found with a Schwann stain, they found that, in the control, they did not find Ki67 protein. On day 3, they found it in the experimental rats, and they noticed that the changes were happening more or less each day. So on day 3, it exhibited signs of the change. In day 5, they saw more changes. In day 7, it peaked. In day 10, it started to decline. And in day 14, it began to stabilize back to normal. And here they used the immunochemistry test to compare the control versus the experimental rats, and they found that each time point, the THY nephritis showed Ki-67. This shows the cell cycle remained active for 3 to 10 days, increasing on days 3 to 5, and peaking on day 7 again, and declining on days 10 to 14. On Figure 1C, the detection of the labeling index was calculated for Ki-67 positive cells versus total glomerular cells on days 3, 5, 7, 10, and 14. And again, they found that the control had no peak in the Ki-67. On day 3, in the experimental rats, they found that they had showed signs of exhibiting Ki-67. On day 5, it continued to increase. On day 7, it peaked. On day 10, it began to decline. And on day 14, it stabilized. Figure 2 shows microRNA 34A. The detection of microRNA 34A gene expression by real-time PCR was used on days 3, 5, 7, 10, and 14. MicroRNA 34A was found to begin to gradually decrease with the progression of the pathological processes. On day 3, being at its lowest point. If you look on N, which is the control, this is the normalized level. On day 3, it began to decline. On day 7, it got to its lowest points. And on day 10, it began to normalize again. And day 14, it went back to normal levels. And this goes along with both the microRNA 34A and the pathological process, both normalized on day 14.
My name is Loretta Granum. I'll be going over Figure 3. In this figure, researchers aim to determine the effects that the microRNA 34A has on the proliferation rate of mesangial cells in the cell cycle. Figure A shows us the optical density of renal mesangial cells over the course of 72 hours. If we look here, we'll see that normal control and the negative control of the microRNA 34A bear similar results, giving us the first indication that microRNA 34A does play a regulatory role in the proliferation of mesangial cells. At 24 hours, there's a reduction, but here, in the presence of the microRNA 34A mimics, after 48 hours, we notice a slightly significant reduction, and we observe even more reduction here at 72 hours in renal mesangial cell proliferation. These results show us that microRNA 34A mimics are influential to the suppression of cell proliferation. Now, in Figure B, we're looking at the results from a flow cytometry method, which focuses on the cellular DNA content. If we look at the normal and control results, we can see that there is somewhat a small range in the DNA context. So if you look here, it's a much smaller range, and an elevated number of cells overall. So that's looking over here. The microRNA 34A mimics, on the other hand, tend to have an extended amount of DNA content, indicating an extension of the GLG1 phase. Consistent with the cells being restricted to the cell arrest or growth phase, we see a reduction in the cell number, meaning that G2 or mitosis phases are also reduced. We're going to take a quick look at Table 4, which shows us further support of the results indicated in Figure 3. By looking at the percentage values consistent with the extension of these phases shown in the previous figure, there is a significant reduction in in the percentage of mesangial cells in the S phase. So if we look here, and we see an increase here in the GO to G1 phase. The decrease here is due to the fact that cells are unable to commit to this particular phase due to being arrested at the restriction point. Lastly, in the G2 to mitosis phase, it further supports the results in figure 3 because we see even less of a percentage of mesangial cells at this point. Being that the cells are not able to proceed past the restriction point in the presence of microRNA 34A mimics to undergo the S phase or the G2 or mitosis phase, growth factors are not binding to their receptors to stimulate cell proliferation.
So, since we've already established that the effects of the MIR34A, we also want to conclude that MIR34A can actually target the platelet-derived growth factor receptor beta. So here is a combined sequence of the mRNA 34A gene and how it can target the growth factor receptor. And it shows this in the 3' untranslated region.
Through dual luciferase assays on fireflies and ranellas, we know that microRNA 34As have binding sites at the 3' untranslated region of the platelet-derived growth factor receptor beta, making it a target gene of microRNA 34A. In order to understand microRNA 34A's effect on the target gene's expression, dual luciferase plasmids were used to monitor the changes in expression of the target gene fused to a reporter protein, side check. This formed a side check platelet-derived growth factor beta plasmid, which was co-transfected with either a microRNA 34A mimic or the microRNA negative control into the renal mesangial cells. And those co-transfected with the mimic, if we look right here, there's a significant downregulation in the firefly luciferase activity. So we can see the big difference here. Okay, so figure B is going to show us the result of real-time qPCR to show that the platelet-derived growth factor receptor beta mRNA is expressed in renal mesangial cells following transfection with either microRNA negative control, microRNA 34A mimics, or in the normal control. This to show that it's expressed, the mRNA is expressed in all. So figure C, we're going to look at the Western blot analysis, which was conducted to observe the protein levels of platelet-derived growth factor receptor betas and the phosphoplatelet-derived growth factor beta, which is the activated form. So as I highlighted here for you. After transfection. So we see that there's a significant decrease in the expression of platelet-derived growth factor receptor beta in its phosphorylated form in the renal mesangial cells that were transfected with the mimics, with the microRNA mimics right here. So we'll see a huge decrease compared to the normal group in the microRNA negative control group. Being that the expression was reduced, researchers were able to determine that microRNA 38 likely targets the inhibition of the platelet-derived growth factor receptor. And we're also going to look at figure D here. We use Western blot analysis was used again to reveal protein expression of platelet-derived growth factor receptor beta and phosphoplatelet-derived growth factor beta, receptor beta in the anti-Thy1 nephritis rat models. Based on the results, day 7 shows the most pathological change. It is on this day that the expression of phosphorylated platelet-derived growth factor receptor is significantly greater, meaning a growth factor is bound. So this is the activated version here. This indicates that platelet-derived growth factor receptor is a key regulator in the ability of anti-Thy nephritis to proliferate. So in figure 5, as we know, the cell cycle requires the assistance of proteins to regulate the progression of the cell cycle. And flow cytometry showed us that microRNA 34A mainly impacts the G0, G1 phase. Therefore, this study focuses on the expression of the GL, G1 phase regulatory proteins and their expression levels. Okay. So here in Figure A, researchers measured the expression levels of the microRNA mRNA, for cyclin D1, cyclin E, cyclin-dependent kinase 2, cyclin-dependent kinase 4, cyclin-dependent kinase 6, and P27. So these are all these proteins here. The results show a significant reduction in the mRNA expression of cyclin E. So if we look here, this is cyclin E, and we'll see it's much lower than everything else. Cyclin E is one of the cyclin proteins needed for the progression into the S phase of the cell cycle by binding to cyclin-dependent kinase 2. Also, in the presence of the microRNA 34A mimic cells, right here, we can see a significant increase in P27, which is a cell cycle protein kinase inhibitor. So figure B, look here, we're looking at the expression levels of the same cell cycle proteins. At this point, we see a significant reduction in the expression of all of these proteins, except the inhibitor, P27, that were exposed under the microRNA 34A mimics. These results are consistent with what researchers hypothesized that microRNA 34A targets the G0, G1 cell cycle proteins. So figures 5C and 5D, there were previous studies conducted to support the idea that cyclin D1, cyclin-dependent kinase 4 and 6, which form their own complex, are target genes. Therefore, in figure C, the focus is placed on, in figure C and D, the focus is placed on cyclin E and cyclin-dependent kinase 2. These are the two proteins that show major reductions in mRNA expression levels when co-transfected with microRNA 34A mimics. So once again, luciferase activity was measured by cloning the 3' untranslated regions of both proteins into sidecheck 2 to form the plasmids sidecheck cyclin E and sidecheck cyclin-dependent kinase 2. Renal mesangial cells were also transfected with the microRNA 34A mimics and the plasmids to determine whether or not the microRNA 34A is a downregulator of cyclin E and cyclin-dependent kinase 2. These results reveal that there's a significant decrease in firefly activity, as we see right here. Therefore, microRNA 34A targets these proteins.
Okay, now we're going to do figure 6. This is in figure A of the 6. The Western blot is showing the expression of these proteins that are related to the RAS MAPK signaling pathway. And they're in the conditions of normal, which would be the regular cells. Con is the miRNA negative control, and the mimics are the transinfected with the MIR34A mesangial cells. So as you can see, RAF1 and ERK2, ERK1 and 2 right here. They show no change, but in MEEK1, it shows a significant decrease. And you can see it as well over in the bar graph. So KRAS and phosphorylated RAS, RAF1 and phosphorylated MEEK1 and phosphorylated ERK1 and 2, they're shown to be down regulated. So in the normal cells, all the levels seem to be even. The con cells show a vary in levels, but not much for the proteins as you're looking in the bar graph. But the mimics have major differences in their expression levels, indicating that for every phosphorylated protein, the levels were low while they were activated. But the significant protein level is the phosphorylated MEEK1, because it barely had any expression, indicating that when the phosphorylated MEEK1 is activated by the MIR34, it's inhibiting the expression. It's also pointed out that MEEK1 is the target gene when being phosphorylated. We'll talk about this part right here. And then for B, when we're looking at the different days, the significance of it was For day 7, it's a distinct visibility that the phosphorylated MEEK1 has increased levels of protein with relation to the expression, but it's not as high as levels for the phosphorylated MEEK1 on day 14. So you can see in the bar graph, this has a major increase as far as staying the same in the first day.
So in figure 7, researchers wanted to confirm the effects of microRNA 34A on the platelet-derived growth factor receptor by introducing a small interfering platelet-derived growth factor receptor on the cell cycle, its related proteins, and the Ras-MAPK signaling pathway to measure its influence. The purpose of the small interfering RNA is it's a tool used in RNA interference techniques to promote gene silencing and targeting of the mRNA for degradation. So, in figure A, we see that renal mesangial cells transfected with the small interfering protein-derived growth factor receptor beta reduce cell proliferation, similar to the effect that the microRNA 34A mimics produce. So here we'll also see this reduction compared to the negative control in the normal. Figure B, we see flow cytometry again, which indicates that small interfering platelet-derived growth factor receptor beta also extends the G0 phase / G1 phase, and it reduces the G2 or mitosis phase here. So for figure C, Western blot analysis is used to show a significant decrease in the cell cycle regulatory proteins, as we saw with the microRNA 34A mimic effects. The results also show a decrease in protein expression of the Ras-MAPK signaling pathway proteins, KRAS, phosphorylated RAF1, phosphorylated MEK, and phosphorylated ERK1 and 2. If we just look here, these are our cell cycle regulatory proteins, and we see the reduction. And then if we look up here, right here it's reduced, and the phosphorylated regions or activated regions are all reduced. So RAF1, MEK1, and ERK1 and 2 did not show a change. So if we look over here in the bar graph, we'll see that these pretty much stayed normal, these 3. This provides evidence that small interfering platelet-derived growth factor receptor beta and the microRNA 34A have similar effects of suppressing the platelet-derived growth factor receptor and the suppression of the cell cycle proteins, cyclins D1 and E, and cyclin-dependent kinases 2, 4, and 6. And it also promotes a significant increase in P27 expression. We can understand that the small interfering RNA and the microRNA 34A directly inhibit the platelet-derived growth factor receptor beta. Then it impacts the Ras-MAPK pathway, followed by the cell cycle regulated proteins. Table 6 shows us further support of the results indicated in figure 7 by looking at the percentage values for the small interfering platelet-derived growth factor receptor beta. We see an increase in the G0, G1 phase, which is consistent with the extension of these phases shown in the previous figure. And there's a significant reduction in the S phase of mesangial cells, since they are unable to commit to this particular phase due to being stuck or arrested at the restriction point. And lastly, we see even more of a reduction in the G2 or mitosis phase, which further supports the results in figure 7 because we see even less of a percentage at that point. Being that the cells are not able to proceed past this restriction point to undergo S phase or the G2 or mitosis phase, growth factors are not binding to their receptors to stimulate cell proliferation.
Okay, next we'll be discussing figure 8. And figure 8 will have the figure 8 will have the cells that were placed in the fetal bovine serum, and inside the what you can see here is that the I apologize. It shows with the different concentrations of the fetal bovine serum that there was a further inhibition with the cells in the further cells that was in the MIR 34A after a 72-hour period. And this confirmed the effects that they see here, that with the application of the fetal bovine serum, that it does have a inhibitory effect on the MIR34A gene and the silencing of the platelet-derived growth factor receptor. Because as previously we thought that we knew that the MRR34A had targeted the platelet-derived growth factor beta, but here when you add 20% of the bovine serum, you clearly have a clear activation of all of the proteins that cascade down for cell signaling.
Exogenous factors of the bovine serum 20% were used to stimulate mesangial cell proliferation to see the effects of microRNA 34A and what it would do to proliferation. We found out that microRNA 34A can inhibit the target gene platelet-derived growth factor function of rat meningeal cells by suppressing G0 through G1 phase. They found was that higher concentrations of serum, of course, accelerated the cell proliferation, but higher concentrations combined with microRNA 34A returned cell proliferation rate to normal levels for 10% serum with a 10% serum culture.
Okay, now that we've gone through all the figures, let's just have a quick recap, shall we? So, as we said in the beginning, that this would be the typical pathway of RAS. You have you have the receptor tyrosine kinase receptor bind from the ligand, that's bound from a ligand, will have further activation from its subunits, and you have the RAS activation. Once it is bound to the GTP, they will further go on to activate RAF, MEEK, will phosphorylate MEEK, activate MAP kinase, and go on to cell transcription. But here, after the further evaluation of the MRR34A, we see that the gene regulates mesangial cell proliferation by directly inhibiting the platelet-derived growth factor beta and the MEEK pathway and the RAS to MAP kinase pathway. This signaling pathway also influences cell proliferation, and it influences the G0 and G1 phase in the cell mitosis pathway by extending this phase and attenuating the S phase and the G2 phase. We also have found that MR34A directly regulates the cyclin E and CD2 in the regulation pathway, which also will cause the G2 and the S phase. This will serve as a better strategy to eliminate tumor proliferation. Thank you, and this will conclude our presentation.